Hello everyone, welcome to my YouTube channel Raj Botany. Myself Dr. Rajendra Balasaheb Kakade, Assistant Professor at Department of Botany, Swami Vivekananda Senior College Manta, District Jalna. Friends, today I will talk on a very important topic, that is structure and function of DNA. Before moving further, we must know the history of DNA discovery or the timeline of DNA discovery. In this regard, Swiss chemist, that is Frederic Michel, in 1860s identified nuclein in the nuclei of human white blood cell, which is also called as leukocytes or WBC. The Frederic Michel was supposed to isolating the protein from the human white blood cell, but he came across the isolation of the nuclein. Then came the Albert question in the year 1881, Nobel Prize winner, German biochemist. He who credited with the naming the DNA, he also identified the nuclein as a nucleic acid. Not only this, he isolated those 5 nitrogen bases, which are now we consider them as a basic building block of DNA and RNA. We know adenine, guanine, cytosine, and thymine. If DNA is there and RNA is there, the earlier 3 nitrogen bases are constant, adenine, cytosine, and guanine. And if the RNA is there, that thymine is replaced by the uracil. So this is a very important difference between DNA and RNA as far as the nitrogen bases are concerned. Then Theodore Boveri and Walter Sutton, who proposed the theory called as Boveri Sutton chromosome theory, also known as chromosomal theory of inheritance. They reported that how chromosomes store the genetic information and transfer that genetic information from parents to their offspring. Then very famous scientist Frederick Griffith, in the year 1928, he performed a well-known experiments. What he did, he took 2 strains of the Streptococcus pneumoniae bacteria, rough strains and smooth strain. Rough was non-virulent, smooth was virulent. He took the strain R, injected to the mice. He found that mouse were healthy. Then in another experiment, he took smooth virulent, injected into the mouse, found that mouse were died. In his third experiment, he took the heat-killed smooth virulent Streptococcus bacteria, injected to the mouse. He found that mouse were healthy one. And in the last and very important experiment, he took the mixture of rough non-virulent strain of Streptococcus and heat-killed smooth virulent strain of the Streptococcus. Jointly, he injected that mixture into the mice and he found that mouse were dead. On the basis of this experiment, he concluded that DNA was the molecule that inherited and causes the conversion of rough strain into the smooth strain that resulted into the death of the mouse. Very important experiment. Then Oswald Avery in 1944 first outlined the DNA as the transforming principle. It's a DNA, not a protein, that transfers the cell properties. Then very famous scientist Irwin Chargaff in the 1940s, he found the base composition adenine, guanine, cytosine, and thymine. He reported that or researched that the amount of adenine and thymine, both composition, is equal to the amount of cytosine and guanine. There should be 1:1 proportion between pairing of purines and pyrimidines. And this is a very famous lady researcher, lady scientist, Rosalind Franklin, in the year 1951, he performed the experiment using the DNA, using the X-ray crystallography. Through this X-ray crystallography technique, one can estimate the structure of 3-dimensional structure of the any biomolecule. So Rosalind Franklin took the X-ray source, bombarded this X-ray on the DNA sample. She observed that the refracted X-rays developed such a pattern on the photographic plate. Such cross bands indicate the helical nature of the DNA molecule. That bands are Very important point is here. These bands indicate the helical nature of the DNA molecules, and this DNA molecule had a constant diameter. These bonds also interfered or also concluded that this DNA molecule has a constant diameter that is 2 nanometer. This is a very what you can say the landmark in case of the DNA structure or the DNA discovery. This is a landmark research that is performed by the lady scientist Rosalind Franklin. Then the scientists came in 1953, Watson Crick. These are the young Watson Crick scientists along with their DNA model, and old Watson Crick. They reported that the structure of DNA is a double helix polymer, and this double helix polymer is a spiral that consisting of the two DNA strands which are wound around a common imaginary axis or wound around each other. And for their discovery, along with the scientist Maurice Wilkins, they shared the Nobel Prize in the year 1962 in the field physiology and medicine. This is a Nobel Prize ceremony. Scientists, different scientists from different countries. Before moving further to the structure of the DNA, we must know the exact location of the DNA. If you consider the case of the human, if you draw the cells of the human, there is a human cell. Inside the cells, nucleus is there. Inside the nucleus, chromosome is there. Here is the chromosome. This is actually the metaphasic chromosome. Here we can see two sister chromatids are there. If we more unwind this metaphasic chromosome, we can see the chromatin fibers, loops of this chromatin fiber. These are the loops of chromatin fiber. This is a electron micrograph picture of this chromatin fibers loops, and these loops are attached on the substratum called as a scaffold, SCAFFOLD, scaffold. And if you more focus on this chromatin fiber, we can see the beaded like structure that is called as nucleosome, bead which are woven into the string. And if we compare that beaded structure to this chromatin fibers, that beads are nothing but the histone which are formed from 8 subunits, S2A, S2B, S3 and H4. All the 4 subunits are in multiple of 2. 4 into 2, 8. Means one histone protein is formed of 8 subunits, and around these histone subunits DNA is coiled. DNA is there, and both successive histone subunits are joined with each other DNA. That DNA is termed as linker DNA. That linker DNA links to histone. That's why name linker DNA. And here is our DNA molecule. So let's see some common point about the DNA. It is a polynucleotide chain which is formed of 2 strands, and these strands are run opposite to each other. DNA is a self-replicating material that's present in every living organism. DNA is a carrier of all genetic information which are required for the development, for the growth, for the survival and for the reproduction of any organism. Most of the DNA is found in the nucleus of the cell, but small amount of DNA is found in the cell organelles like chloroplast and mitochondria. Such DNA is called as extrachromosomal DNA or cytoplasmic DNA. Nearly every cell in our body has the same DNA. Now let's see the definition of the DNA. It is a type of nucleic acid which is made up of the sugar, phosphate and nitrogenous bases. Type of nucleic acid means there are 2 types of nucleic acid, RNA and DNA. DNA is a type of nucleic acid which is made up of sugar, phosphate and nitrogenous bases, and polynucleotide chain which stores and transfer the biological information from parents to the offspring. Such biomolecule is called as DNA, deoxyribose nucleic acid. Let's see the general structure of this DNA. It is a general structure of a DNA molecule. It is a double helical molecule consisting of the two strands which are running anti-parallel to each other and which are also piled around each other around an imaginary axis in a right-handed fashion. In a right-handed fashion, and this DNA molecule is a polar in nature, means it shows 3 prime to 5 prime. What you can say, the arrangement, and another stand 5 prime to 3 prime, means DNA is a polar in nature. DNA also show major groove and minor groove in an alternate manner. Here we can see along the entire length of this DNA molecule, major and minor grooves are there. Here major, minor. So here major, minor, again major. Okay, so what exactly the role of this major and minor groove? These grooves are important in the attachment of the DNA binding protein in the process of replication and transcription. During the process of replication and transcription, DNA binding proteins are attached at this minor and major grooves. This major grooves is deep and wide. Here we can see it is deeply situated, whereas minor groove is shallowly situated. Let's see the dimension of this DNA molecule. The diameter of this DNA is 2 nanometer or 20 angstrom. Length of one helix, we must know what exactly the one helix means. Distance from this to this is called as one helix, and the length of this one helix is 3.4 nanometer or 34 angstrom unit. The distance between these two successive rungs, if you compare this DNA molecule with the ladder, then the distance between this and this rungs is 0.34 nanometer or 3.4 angstrom unit. Therefore, each one helix have 10 base pairs. Let's see the chemical composition of the DNA molecule. DNA is a long molecule that is made up of repeating individual units of monomer called as nucleotide. This is structure of the nucleotide, which is made up of 3 basic components. Very important point here. The nucleotides composed of 3 basic components. First, sugar base, that is nitrogen base, and phosphate. At the first carbon atom of this sugar, nitrogen base is attached. At the third carbon position of this sugar, hydroxyl group is attached. And at the fifth carbon, which is normal present outside the ring, at the fifth carbon of this sugar, phosphate group is attached. Let's see detail about the deoxyribose sugar. Its molecular formula is C5H10O5. This ring having the pentagonal structure, and this sugar along with the phosphate forms the backbone of the DNA. Here this is a sugar, phosphate, sugar, phosphate, sugar, phosphate. Along with the phosphate, this sugar forms sugar phosphate backbone of the DNA molecule. In this ring we can see 1, 2, 3 and 4 carbons are present in the ring, and fifth carbon present outside the ring. And at the carbon number first, third and fifth, hydroxyl groups are there, whereas at the carbon number second, no hydroxyl group, only hydrogen is there, oxygen is lacking. That means deoxyribose sugar. Name is given deoxyribose sugar present in the DNA, whereas in RNA ribose sugar is there. Why? Because at the second position also hydroxyl group is present. The sugars of the two strands are directed in opposite direction. If you see this figure, this is sugar, and this sugar are arranged or oriented in the opposite direction as compared to the orientation of these sugars. Now let's move further to the phosphate group. The phosphate group, actually the phosphoric acid molecular formula is given S3PO4, that having the 3 active hydroxyl group, first, second and third. Out of these 3 hydroxyl groups, 2 hydroxyl groups are actively involved in the strand formation, DNA strand formation. In the DNA strand, phosphate group is alternating with the deoxyribose. You can see phosphate, sugar. And if you more concentrate on these phosphate group, here we can see, as far as this sugar is concerned, at the third carbon of this sugar, the oxygen of this phosphate is attached, and at the fifth carbon of another sugar, oxygen of this phosphate group is attached. Means third carbon of the one sugar and fifth carbon of the another sugar, phosphate group form the ester bonds. Two ester bonds are there. That's why phosphodiester bonds are there in the DNA molecule, and these bonds are formed by the sugar and phosphate group. Okay, thus each strand has 3' end and 5' end. Here we can see 3' end, means at the 3' end OH group is there, and at the 5' end phosphate group is there. Here also in this strand 3' end, at the 3' end OH group is there, and at the 5th dash end or 5' end phosphate group is there. Two strands are, here we can see two strands are oriented in opposite direction. One strand is oriented from 5 to 3, another from 3 to 5, opposite orientation in between two strands of the DNA. Then let's move toward the nitrogenous bases. Two types of nitrogen bases are there, purines and pyrimides. Here we can see the purines are the what we can say the double ring structures. Adenine and guanine show the double ring structure, whereas pyrimides are single ring structures. And these nitrogen bases form the steps in the DNA molecule. Here we can see the nitrogenous bases which are attached with each other by this hydrogen bond. If guanine and cytosine is there, 3 hydrogen bonds are there. If thymine and adenine is there, 2 hydrogen bonds are there. And these purines and pyrimides are the actually aromatic heterocyclic compounds which are weak basic in nature. And these purines and pyrimides are hydrophobic in nature. And these nitrogen bases, very important point, these nitrogen bases also follow the Chargaff's rules. That means A plus G is equal to T plus C. Next, that is a bonds in DNA. If you see the bonding pattern along the DNA structure, first very important one that we discussed earlier, phosphodiester bond. This phosphodiester bond is formed by condensation reaction. Here we can see at the third carbon of one sugar, there is a oxygen is attached to the carbon atom, third carbon atom of this one sugar, and at the fifth carbon of this another sugar, oxygen atom of this phosphate is attached. Two ester bond is there, one is here, another is here, two ester bonds are there, and the bond is named as phosphodiester bond. Okay, very important bonding pattern. Then hydrogen bonds, these hydrogen bonds are formed in between purines and pyrimides. These hydrogen bonds is actually temporary bonds, but they stabilizes the DNA molecule. In between guanine and cytosine, 3 hydrogen bonds are there, 1, 2 and 3. And as far as adenine and thymine is concerned, 2 hydrogen bonds are there. Glycosidic bonds. These bonds are formed between sugar and nitrogenous bases. Sugar, first carbon of the sugar and nitrogen of this nitrogen bases, there is a formation of this glycosidic bond. Here is glycosidic bond. Here is also glycosidic bond. If you consider this strand, here is glycosidic bond. Here is also glycosidic bonds. Means 3 main bonds are there in the structure of DNA: phosphodiester bond, hydrogen bonds, and glycosidic bonds. Let's see the DNA function. DNA contains all the genetic coding which is used to control the functions, behavior, and development of an organism. DNA is also used as a long-term storage device. That means DNA store the genetic information, and that genetic information gets transferred from one generation to the next generation. DNA is majorly function as far as the protein synthesis is concerned, because from DNA mRNA gets formed by the process called transcription. DNA encodes on the mRNA, and this mRNA after formation come into the cytoplasm through the nuclear pore, and into the cytoplasm further process of translation get happen and protein get formed. Then DNA undergo mutation That means the permanent changes that passed on to the offspring, and this mutation causes the evolution among the species. DNA also have the capacity of self-duplication or replication, or it can inherit from parent to the offspring. If you know the exact chemistry of the DNA molecule, then we can manipulate it for the benefit of human. Let's say, for example, if genetically modified crop that are pest or drought resistant that have been generated from wild type varieties for the genetic engineering. Means if you know the chemistry of the DNA molecule, then we can develop disease and pest resisting variety of the crop. Thank you viewers for your patient sharing. I request all of you, if you like this video, please hit the like button. Please subscribe my channel if you not subscribed it, and do not forget to press the bell icon for the updating video. Thank you very much.